Good morning, everybody. Welcome to the National Institute for Occupational Health COVID-19 online webinar as one of the many webinars we've had in the series of the COVID-19 training and capacity building webinars. This particular webinar on COVID-19 vaccine and vaccinations is a repeat of a webinar we did on the 4th of March this year, and by popular demand, we have been requested by the NIH stakeholders for a repeat of this particular topic, and we are very, very thankful to our list of distinguished speakers and presenters for availing themselves, given also the quite busy schedules they have within the Sosonke Open Trial, as well as the Gauteng Department of Health and Steve Biko Academic Hospital and the Immunology Department at the Charlotte Matreke Johannesburg Academic Hospital. So thank you to our speakers. And yes, this is a very important topic, vaccines and vaccinations within the COVID epidemic within our country and the pandemic globally. And it's very important for all of the occupational health, occupational medicine, occupational health and safety practitioners in the workplaces in South Africa to be fully informed and to be able to make all of our contributions towards strengthening and preparing for the ongoing challenges we face, and particularly strengthening the occupational health and safety preparedness, COVID-19 preparedness, and prevention within the workplaces. We all have very important roles to play, obviously from those in charge of our workplaces, from the health and safety representatives, COVID-19 compliance officers, the occupational medical practitioners, the doctors and the nurses who play an occupational health and occupational medicine role within our workplace, and all the other specialists, hygienists, etc., environmental health practitioners, as well as employee health and wellness practitioners within our workplace. So welcome again to the NIH's online COVID-19 webinar. The NIH is a specialized division of the National Health Laboratory Services and a sister division of the National Institute for Communicable Diseases. And without much ado, and to give as much time to our colleagues as presenters, I will be introducing to you the program and our presenters, and then move over to our first speaker. But just quick admin with regard to our virtual webinar. For the participants, your microphones and sharing of any files and the hands raising functions have been deactivated. Please do not use that. Please type all of your questions in the question and answer box. That's the Q&A box, so that our presenters and panelists can answer your questions there live in the Q&A box. For the chat box, it's general announcements, general comments, and I will be posting a few important messages and information there for you. So our program contains the COVID-19 vaccine science, development, and safety. And that topic by Dr. Simba Takuva is joined by his colleague from the Sosonke Open Trial. And the second topic that is part of a package is the COVID-19 workplace program considerations by Dr. Azwi Takalani, and they both safety physicians and medical monitors at the Sosonke Open Trial. Dr. Takuva is is a lead safety physician. The third topic is COVID-19 vaccine for workers, and this is an occupational health perspective, which is provided by Dr. Sipho Senabe, who's the Chief Director and HRG and Employee Health and Wellness within the Gauteng Department of Health. Our fourth presentation is an occupational health-based clinical trial in vaccination. And this is an occupational medical practitioner's perspective on the ground by Dr. Lumka Puwani. And she's based, as I said, at the Steve Biko Academic Hospital, when I mentioned the institutions contributing today. And then finally, on the topic of vaccines and SARS-CoV-2, all of the key questions that have been raised in the recent period and months, and questions about how do we proceed going forward, is covered by Professor Elizabeth Main, who is the NHLS, the National Health Laboratory Services Head of Department of Immunology at Charlotte Matete, Johannesburg Academic Hospital. And that's our program for today. I'm now going to immediately hand over to Dr. Simba Takuva, who is the lead safety physician, medical monitor at Sisonke Open Trial. And whilst he prepares to share his slides, I'm going to just briefly do a bit of an introduction. As I said, Dr. Takuva and Dr. Azwita Kalani are both from Sisonke Open Trial, the open label trial, apologies. And the two of them will be presenting from the very practical experience that happens there. So Dr. Takuva, as well as Dr. Takalani, work as medical monitors and safety physicians with the COVID-19 Prevention Network and the HIV Vaccines Trial Network, the two networks, operations in sub-Saharan Africa. They both are involved in the HIV vaccine trial networks that conducts all phases of clinical trials, from evaluating experimental vaccines for safety and immunogenicity, I hope I pronounced that properly, to testing vaccine efficacy, leveraging on the infectious disease expertise of the existing research networks and global partners. The NIAID formed the COV-PN, that is the COVID-19 Prevention Network, whose mission is to conduct phase 3 efficacy trials for COVID-19 vaccines and monoclonal antibodies. Both Simba and Azwi, their first names, will discuss COVID-19 vaccine science, development, safety, as well as COVID-19 vaccine operational program considerations. So in a way, I've introduced both our speakers 1 and 2. I hand over to you, Dr. Takuva. Thank you very much again for joining us today and sharing your knowledge and experience with our target audience. Please proceed. I'm not sure if you need to unmute your microphone, Dr. Takuva. There we go.
Thank you, Ashraf. Thank you, Ashraf. Your pronunciations are quite on point. Thank you. My role will be to give a really general overview of COVID vaccines, to look at the type of vaccines that are there, the clinical development, and I'll spend a bit more time looking at safety and also some of what we're seeing in Sisonke. The presentation is really preached so that one does not need to have a deep understanding of immunology or vaccinology to understand. So hopefully, yeah, others will also feel the same, that it's a gentle presentation and they are able to understand. So that's the outline. I think I've spoken to it already. And just as a potential conflict, yes, we work in COVID-19 and HIV vaccines, and some of the trials use industry products. So the first question really is, why do we need a vaccine, and what could the vaccine do if it is safe and effective? So this is always useful if one looks at it at two different levels. The first level being at the individual level. What will it do for you as an individual, for you and me as individuals, really? So first, what we're already seeing with some of the data from the COVID vaccination trials is that these vaccines are quite effective in reducing the severity of illness. So meaning that even if you get COVID, you're less likely to have severe illness. You might be symptomatic or less symptomatic, but you're less likely maybe to have a higher risk of critical illnesses, hospitalizations, or even death. So this is quite important because then it reduces morbidity and mortality due to COVID. And then also another potential benefit is that the vaccines are likely also to prevent infections, so preventing infections within individuals. So this is data that is still emerging, and there is suggestion that for sure the COVID vaccines might be effective in preventing infections amongst individuals. And then when you go a step higher, now you're looking at it at a community or a group level. So how can a COVID vaccine benefit us as a community or us as a group? So when you look at the vaccine preventing infection in individuals, so this translates to reducing transmissions or reducing onward transmissions from person to person. And we've all heard about the concept of herd immunity. So this is a threshold that if achieved, there's likelihood that you actually cut off the transmission of an infectious agent to below your reproductive levels, where it's able to continuously propagate in the community. And that way you then are able to have a community that can have either minimal or even free of an infectious disease. And for instance, this number for South Africa has been put forward as if we are able to vaccinate 70% of the population, there is a good chance to actually achieve that herd immunity over time. So that's one benefit of a COVID vaccine when you look at it at the community level. So when you then extrapolate those benefits, this then leads to healthier communities. People are less sick. People are able to, I know this is an occupational health platform. People are then less likely to file in more sick days. There's more productivity at work, etc. And then overall, when you look at the health system, then there is less stress on the health systems. I'm sure most of us will admit that currently it seems like, it looks like the focus has really been on COVID, and there is concern that perhaps we may not be paying much attention to the diseases that have always been there and that have always been giving communities a hard time, as quite a number of resources and quite a number of focus has moved on to COVID. So by eliminating COVID or controlling COVID, there's then less stress on the health system. Then how does a vaccine work? So this is a cartoon that aims to illustrate how a vaccine could potentially work. So a vaccine stimulates the human body's own protective immune responses, so that if a person is infected with a pathogen or a germ, the immune system can quickly prevent the infection from spreading within the body and causing disease. In this way, a vaccine is actually mimicking natural infection, but without actually causing the person to be sick. So if you look at that cartoon, it says a vaccine works by teaching the body to recognize and fight invaders. For instance, you give a vaccine that contains components that maybe look like COVID, or that the body thinks it's been attacked by COVID. The body then sounds that alarm where it calls into action different components of its arsenal. So this is your fighter cells, your antibodies, etc. And then they go into action, and then they fight and neutralize or block or control that virus from then propagating or spreading further in the body and causing disease. So in order to better have a sense of really how COVID vaccines work and how they are designed, it's always important to take a step back and understand the different parts of the virus that are used. And understanding the different parts of the virus that are used, it's important to have a sense of how a COVID virus actually looks like. So if you see the picture on your extreme right, so that's here. So this is a schema of the COVID, a simplified schema of the COVID virus. So you'll see start off like as a maybe spherical molecule or organism, and then on its surface, it's got the spikes. So the spikes known as the spike protein. So most vaccines focus on the spike protein of the virus. So the immune system is trained to develop antibodies that then latch onto the spike proteins. And by latching onto the spike proteins, they're then blocking the spike proteins from attaching to the human cell. As you'll see in the next slide, how then COVID infects a human cell is that those spike proteins attach to the surface of a human cell, and via some receptors, this then allows the COVID to actually infect the human cell. So you then actually develop, so by giving a vaccine that actually contains a component of the spike protein, you train the body to actually be able to fight that part of the virus so that the virus is unable to then infect the human body. So let's move on to the next slide, which again is another schema that aims to illustrate how this happens. So again, if you look at your top picture there, you've got a COVID virus, and then you've got those orange spikes. Those are the spike proteins. Then it comes in contact with the human cell. Then when it comes in contact with the human cell, what ordinarily would happen is that it then uses some of those receptors on the surface of the human cells, like your transmembrane ACE2 receptors, to then infect and get into the cell. But what then your anti-spike protein antibody, which would be triggered or which will be produced when you have an effective vaccine and it produced through an antibody response, what it then does, those Y-shaped molecules that are known as antibodies, they then block those spike proteins. Think of them as landing docks. They block them, and by blocking them, then the virus is not able to infect the cell. So this is just really one component of the immune system, but there are other components of the immune system that come into play as well. So if you think of other responses like your T cell responses, etc. And then, now that we have a sense of how a COVID virus looks like and how a COVID vaccine could work, let's now look at the pieces of the vaccine. So one could think of a vaccine as a sandwich. So it's made up of different pieces. So what we've been speaking about in the previous slide is really what we'd call the antigen. So this is what you actually use to actually trigger that immune response. But when you design a vaccine, you just do not give an antigen. You have to construct something that's more complete, and that's for different reasons. So one way to look at it is to think of it as a sandwich. So you could have a sandwich whereby you actually have a genetic code, and in some vaccines, this is the recipe for the sandwich, and that is all that is needed. So a genetic code is a set of instructions. So this is a recipe for the sandwich. So the genetic code in a vaccine gives your body the instructions necessary to create the antigen that your immune system will respond to. And then if you move on to the next one, it says vector there. So a vector is a carrier and is used to carry the antigen, like the bread that helps you carry the sandwich filling. Because to have a sandwich, those fillings inside the sandwich, you need the bread, right, to actually carry the sandwich fillings. So these vectors are often other viruses that are not harmful in humans or that have been made replication incompetent. And there are many different kinds of vectors, just like you can choose between you know, there are fancy breads nowadays. When you walk into the supermarket, it's no longer like, when I grew up, it was white or brown, or maybe it depends where one grows up, but it was just white or brown bread. But now you've got wheat bread, you've got rye, you've got sardoff, you might have croissants, you might have a hamburger bun. So there are different vectors as well. And then the antigen, this is what I've spoken about in the previous slide when we spoke of the spike protein. So if you look at most of the vaccines, really the antigen is the spike protein. So this is the main ingredient inside the sandwich, such as the meat, the cold meat, etc. And this is what our immune system will be responding to. And in some vaccines as well, they have other components known as adjuvants. So think of this as like maybe your sauce or your tomato sauce or your mustard that you add to your sandwich so that it's more tastier, more palatable. So the adjuvant is added in some vaccines, and it's used to boost the body's reaction to the vaccine. That is like give it an extra kick. So that well, thinking of it in the sandwich analogy, it's so that your food is tastier, right? Yeah, so some vaccines actually need an adjuvant so that they have a much more robust and complete immune response. An example being like if you look at the Novavax vaccine, but we'll look at some of the examples of vaccines after we speak about the actual clinical development of vaccines. So vaccine development normally occurs sequentially. So you're looking at ideas coming up among scientists published in papers, then there's research that is in labs, in academic institutions, and then the best ideas then move forward to animal testing. You're looking at different levels of animals, and then if these ideas pass through animal testing and the compounds are found to be safe and maybe able to do what they're supposed to do at a molecular level, like for instance, trigger immunogenicity, etc., then this move on to the human studies. Then in the human studies, again, it's usually sequential, and this is in different phases, like from your phase 1, where you're actually starting to look at like safety, and then you're trying to find like dose, the pharmacokinetics, understand the pharmacodynamics of a molecule. And then later on, it moves on to another phase where preceding that, certain criteria need to be met and the vaccine needs to demonstrate certain success before it moves on to the next criteria, such that by the time you get to phase 3, you're now having multi-center studies that are large and you're recruiting thousands of participants from different facilities, and you try to confirm effectiveness and also monitor side effects. And at this phase, if your vaccine works, you're more likely to actually then apply for licensure. And after licensing, what then happens is that you don't stop monitoring for safety, you still continue monitoring for that safety. And then you're now also looking at things like the benefit risk ratio of continuing to roll out that vaccine. But what has happened with the COVID vaccine trials, as you can see with traditional development, it's been sequential. And if you look at it, if you do it sequentially, you're looking at time periods of like 10 to 15 years or longer. But with COVID vaccine development, these have been done in parallel. And as you can see, this actually reduces the time in which you can actually develop your products or vaccines without compromising safety or without compromising any of the components that you use or any of the components that you measure or take care of during sequential development. And this is also facilitated by the regulatory authorities being able to review applications on a rolling basis. So again, this is a reminder. What I'll do, because I see time is like flying, I would really want to focus on safety, but I would like to remind you again, we spoke about those sandwich components. This leads us to the 4 main different types of vaccines that are currently available. Some can further break down those different types into like 6, 7, 8 types, but broadly there are 4 types or 4 categories. Like the first category, it's like your inactivated whole vaccines, whereby a chemically killed virus or subunits of the virus grown under controlled conditions are then given or are then shown to the immune system, and then the immune system then develops a response. So common vaccines such as the smallpox vaccines or flu vaccines use the whole or weakened form of the virus. Some vaccines that are already being rolled out in Africa, like for instance, if you look at some countries in Southern and Eastern Africa, they use this approach. So an example is the Sinopharm vaccine that has been rolled out recently, I think that Zimbabwe, Malawi, and other parts of Southern and East Africa as well. Then the next approach is your viral vector. We've spoken about the vectors when we looked at the sandwich. We spoke of the bread, right? That sort of like carries your pieces of the sandwich. So your viral vector, these are chemically weakened or genetically modified viruses that carry DNA or genetic material that contains specific sequences that code for the antigen. So examples are, for instance, the AstraZeneca vaccine and the Johnson & Johnson or J&J vaccine. And then you've got your nucleic acid approach, and you've heard about this a lot, I'm sure, in the news, your mRNA vaccines, the ones where scientists are always reiterating that they do not change your genetic composition or affect your genetic material per se to change it. These are genetically engineered plasmid containing vaccines that have genetic sequences in them that code for the specific disease antigen. And the ones that are currently being used globally and probably soon in South Africa as well, it's the messenger RNA type. And examples are like the vaccines from Moderna and Pfizer. And then last but not least, there's the protein subunit approach. So these are pieces or fragments of protein from the virus or from the antigen that are used to trigger an immune response. Same as you would do like with the hepatitis B vaccine. And an example, for instance, would be the Novavax vaccine, which has been trialed in South Africa. And these are some of the vaccines really that have been available globally. But just to illustrate, if you look at your extreme left in terms of effectiveness, your flu vaccines, you're looking at efficacy ranging or varying from like 90% to 60%, depending on year. And then when you look at the COVID vaccines, you're looking at efficacy varying from like just above 65% to as high as 95%. But just to reiterate, it's not always fair to compare these vaccines in terms of efficacy. You cannot really say a vaccine that has 95% efficacy is therefore better than a vaccine that has 70% efficacy. There are a lot of things that you have to take into consideration. It's not just always about efficacy. You need to think about the environment where the vaccine is tested, and you need to think about the hosts as well. So on who is receiving the vaccines and what are the host characteristics, etc. And then, as Azu will speak to later, you need to think about other operational considerations as well. And then on your extreme right, it shows some vaccines that have very high efficacies that are currently in our EPI schedules, like your rubella and your polio vaccines. So this is just data showing effectiveness. The Pfizer vaccine, which might be landing on our shows soon for the rollout. And then another vaccine that is currently in use in South Africa via a phase 3B trial is the Johnson & Johnson vaccine, which is a viral vector. So this is a vaccine that has shown 85% efficacy against severe COVID-19 globally across different countries. And it's really been consistent in showing this. And importantly, it's shown equally high protection in South Africa, where we have the B1351 variant that is circulating. And when it was tested, the variant was quite prevalent. Greater than 95% of the isolates were of the variant. So we know it's highly effective against the currently circulating variant that we are aware of. And it really showed complete protection against the most severe of disease. So that's your critical illness resulting in hospitalizations and deaths. Then I'm sure you've seen this cartoon, especially on social media. It's all over the place. So this compares the COVID vaccines according to logistics, etc. But one thing to highlight is if you look at this vaccine, some of the differences are in terms of storage. So if you look at your viral vector vaccines like Oxford, AstraZeneca, Johnson & Johnson, Sinopharm, the whole or in a attenuated approach, or the gamma layer, which is a viral vector as well. These have storage conditions that are compatible with normal refrigeration over short periods of time or reasonable periods of time. And then when you look at your mRNA vaccines like Moderna, Pfizer, you're looking at more stringent storage conditions, like you're looking at -20 degrees Celsius or -70 degrees Celsius. And then most of those are 2 shots, but then when you look at the Johnson & Johnson vaccine, this is a single shot. But importantly, how safe are those vaccines? So this is data that comes from VSafe, which is the CDC FDA Vaccine Adverse Event Reporting System. So this is data from mid-February. There's been more data that has come in, but in a nutshell, it really demonstrates that the adverse events that have been seen are adverse events that have been expected, and there really hasn't been much adverse events of concern. So it's really been consistent with what we've seen in clinical trials. Like for instance, reactogenicity. Reactogenicity, when you get a vaccine, what then happens is that you develop an immune response to that vaccine. And in some people, it might manifest phenotypically with signs and symptoms that might look like a flu-like illness, like having pain, fatigue, headache, myalgia, chills, fever, swelling, joint pain, etc. And I'm sure if you've vaccinated an infant, there's always that, I wouldn't say concern, but there's always that watch out, like when you go back home to say, are they going to be irritable? Are they going to sleep well? Are they going to have a fever, etc.? That's a lot of the time is reactogenicity, and that is expected with any vaccine. And this has been seen really. But then when we look at the most severe or scary adverse events, well, like for instance, if you think about anaphylaxis, which is a severe allergic reaction that is potentially fatal, especially if it happens outside the hospital, these have been really, really rare. So you're looking at a rate of about 2 to 5 per million doses. And I can confirm that with the Sisonke program, we have not yet seen any anaphylactic reaction or any reaction that is consistent with the standardized case definitions for anaphylaxis. So this is some of the safety data from the vaccine that is used in Sisonke. This is from the clinical trial. It actually showed that it demonstrated an acceptable safety and reactogenicity profile. Most adverse events were mild or moderate, and they generally resolved within 1 to 2 days post-vaccination. And this is the picture that we're also seeing in Sisonke, really. And this platform, even before the phase 3B Sisonke program began, had already been administered to over 200,000 individuals. So this is like either as a COVID vaccine or as a vaccine for other therapeutic areas like Ebola, your filoviruses, your RSV, etc. And then when you look at your reactogenicity profile, similar to what we're seeing in Sisonke, people do complain of pain, sometimes some redness on the injection site, swelling. Then in terms of systemic adverse events. Some may complain of fatigue, headache, muscle weakness for a while, joint pains, maybe nausea, and quite a number of fever as well. But these are usually self-limiting and resolve within 1 to 2, sometimes up to 3-4 days or so. And then when you look at your serious adverse events, in the trial, they were really balanced across placebo and the vaccine arms, suggesting that the vaccine did not confer any increased likelihood of serious adverse events. But then when you look at COVID-related deaths, the vaccine showed really high efficacy in reducing COVID-related deaths. So as a reminder, when one is vaccinated with the COVID vaccine, it is normal to experience side effects during the first few days, about 1 to 2 days after vaccination. It's a sign your body is building up immunity, and you may experience on the site or on the arm where you got the injection, pain, swelling, redness. Usually this goes away on its own. If it doesn't, sometimes pain medication like paracetamol or Brufen or others might actually help, or applying a cool wet cloth over the injected area. And throughout the rest of your body, you might feel feverish, experience chills, maybe a slight headache, tiredness, some joint aches, and these are usually self-limiting. We always encourage that if participants that have been vaccinated experience any of those symptoms, they need to report to Sisonke. So there's a toll-free number that's on the screen. It's also available at the back of the vaccination cards. And after you've been vaccinated, you also receive an SMS with an AE reporting link where you can electronically report. And what the safety team does is that if your symptoms are of concerns, or if it's symptoms that look like they need further attention, they then get in touch with the participants and follow them through. And then in terms of allergic reactions, we haven't seen anaphylaxis, but allergic reactions do occur, and a few have been seen, like where you have participants that already have an allergic predisposition and have been reactive to maybe previous injections of vaccines. There are a few allergic reactions that have been seen, but they've really been successfully managed within the health facilities. So what usually happens is that after vaccination, there's a 15-minute observation period. And if you've got an allergic predisposition or an atopic predisposition, or if the healthcare workers think you're at high risk of allergy, your observation period is extended to 30 minutes. And we encourage people that actually come for vaccination to disclose this information upfront, because it's really important. And also what's important is that being vaccinated does not mean that you're immune from COVID. So if you experience any signs and symptoms that might be consistent with COVID, please report them. Please report them. We're going to go over this, but maybe we'll do it at another time. We'll share the slides, but this is just to show the sophistication of pharmacovigilance or tracking of safety events in Sisonke. Yeah, so safety events are tracked, are followed up, but the system is highly passive, meaning that it relies on vaccinees to actually report those adverse events. However, for the more serious adverse events like COVID or deaths related to COVID or hospitalization, the system is built to be more active, and this will be implemented soon. And last but not least, this is a snapshot of some of the adverse events that we're seeing in the Sisonke trial. So among those that have reported an adverse event, most really reported minor systemic reactions. About half of those that were reporting adverse events reported minor systemic reactions. So this is minor fevers, minor pains, general aches and pains, sometimes joint pains or muscle aches. And then in terms of your minor local reactions, among those reporting adverse events, close to 1 in 4 or just above 1 in 4 reported minor local reactions. So that is pain on the injection site, maybe redness, a little bit of swelling. And then when you look at your severe local reactions, like for instance, having pain that lasts for more than 3 days or some induration, that is persistent. This has been just above 5%, and your severe systemic reactions, it's been about 15%. And this has really been driven by people experiencing what we define as severe fevers. So that's your fevers greater than 38 degrees C, etc. And for your serious adverse events, there have been cases of allergy, but none of that allergy has been classified as really severe or meeting the definition for anaphylaxis. We are starting to see COVID disease. We are also seeing some adverse events of special interest. And there have been also unrelated mortality, not related to the vaccine, but due to other illnesses as well. And then in terms of recovery, participants that have been followed up for recovery, close to 96% have confirmed that they've recovered, and we have just below 3% that mentioned that they have not recovered, and we are following up with them to see, like, could it be that they've got other comorbid illnesses, or is there exacerbation of current illnesses as well? But in closing, as of today, greater than 480 million COVID doses have been administered globally. So if you were waiting to see if it's safe and effective by seeing what happens, no need to wait anymore. About 500 million have been vaccinated already. And overall, the safety profiles of COVID vaccines are reassuring and consistent with that observed from the pre-authorization clinical trials. Continue to stay safe from COVID-19, observe social distancing, sanitize, wear a mask, and importantly, when it's your turn, get vaccinated. Thank you for allowing me to speak to the COVID science, development, and safety. I've eaten into Azra's time. My apologies for that. I'll hand over to you, Ashraf. Thank you.
Thank you very much. That's Dr. Simba Takuva, lead safety physician and medical monitor at Sisonke Open Trial. My sense is that you and Dr. Takalani are a tag team and working there in as a team. So I'm sure she will not have a problem with the few minutes you've taken. And just to say that I've had my jab as an NHLS staff member, as an NIH staff member here at Charlotte, and I will encourage everybody to ensure that once the opportunity arises, that they should definitely take it. If they've not yet registered, if they're required to do so on the EVDS system. And by way of introduction, I think I've already indicated that there is a team effort there between Dr. Takuva and Dr. Takalani. And again, Azvi, as her first name is sorry, Azvi is also part of the medical monitors and safety physician at the COVID-19 Prevention Network, and as well as the other network, the HIV Vaccine Trials Network. And I've given you a sense of what those two networks are engaging in and how they've originated and the relationships there. And so the second part of discussing the vaccine science development and safety is being covered now, with specifically looking at the COVID-19 vaccine workplace program considerations. And I immediately hand over to Dr. Azvi Takalani. Thank you very much for making time to join us in your busy schedules. And I know you have been out in the field, and but we needed you to stick to your laptop for an hour or so for us to share your experience and knowledge with our audience and our stakeholders. I hand over to you, Dr. Takalani.
Thank you very much, Ashraf, for inviting us back to talk about this very important topic. I think what Dr. Takuva has taken us through is really the core of this discussion, because all workplace program considerations really hinge on understanding the effectiveness of the vaccine, its safety profile, and actually what goes into the development of a vaccine. So my presentation is going to be a lot briefer, just really highlighting what some of those considerations should be as we as occupational healthcare practitioners start looking into developing or designing these workplace-based programs. So again, before I start, I also do want to declare that I am a safety physician and medical monitor on ongoing COVID-19 and HIV vaccine trials. And the majority of these trials are either sponsored or partnerships with industry. And I am also particularly working on the Sisonke open label trial, which is vaccinating healthcare workers with their Johnson & Johnson products. Okay, so in terms of my considerations, I'm really going to outline my presentation break it down into two phases. So considerations that occupational health practitioners should be thinking about in the preparation phase, which is right now, before we actually start vaccinating people in workplaces. And the second part of the considerations is really things that we should be thinking about now, but are really going to come into effect once we have started vaccinating people in the workplace, and those that will be that I've captioned under the operational phase. So in the preparation phase, I think it's first important to go back to the National Department of Health and its phased approach for vaccine introduction into the population of South Africa. And as we all know, it's divided into three phases, of which phase one is really frontline healthcare workers. And this is set to begin very soon. And currently we are trying to bridge this gap via the Sisonke open label trial in terms of offering healthcare workers early access to vaccination as a phase 3B trial. But what is going to become very important when we move into phase 2 and phase 3 is that the workplace is going to become, or is already enshrined as one of the pillars in the plan of the National Department of Health rollout plan. And so when we're looking particularly at phase 2 and vaccinating essential workers, and also some of our workers that may fall into the high-risk categories, then we know that as occupational healthcare workers, we're going to have a big role to play. So that role is not yet outlined specifically, and probably will be receiving details in the next coming weeks as phase 1 comes into play and we start preparing for phase 2. But what we do know is that vaccinations will be taking place in public health facilities, in vaccination centers, as outreach programs, so like in mobile clinics, and lastly, but most importantly for this audience, is work-based vaccination programs. And this is particularly suitable for essential workers, for example, workers in the mining sector, industry, and the various departments. So when we start thinking about building a work-based program, I think the first thing that we would all like as occupational health practitioners is to optimize coverage in the workplace. So vaccinate as many employees as possible, because we know this will ultimately be very beneficial for not just the employees, but the company and the workplace health and the workplace in general. So the first question that I just really want to answer up front, which comes up often, is that will we be able to just put in place mandatory vaccination policies in the workplace and sort of like force employees to be vaccinated? If you are not vaccinated, then you can't come to work or show a pass in order to continue working. So if we look at the current legislative framework of South Africa, mandatory vaccination policies will probably not be implementable as a blanket rule. There may be exemptions here and there where people may be more inclined to vaccinate, or where the push to be vaccinated may be more heavier than in other places. But generally for the various workplaces, for the general workplaces, we're not going to see mandatory vaccination places coming into play. And this is essentially, if you look at the Constitution, if you look at Section 12 of the Bill of Rights, which talks about freedom and security of the person, it states that everyone has the right to bodily and psychological integrity, and this includes the right to security in and control over their bodies. And if you come to the National Health Act, and you look at number 61 of 2003, and you look at section 7, which talks about informed consent, it states that a health service may not be provided to a user without that user's consent. And if you particularly look at the utterances of the custodian or the current custodian of the National Health Act, which is Minister Zweli Mkhize, when briefing the Parliament at one point, he said the vaccine will be voluntary, but we try to urge as many as possible to have the vaccine. Vaccines are not mandatory. We just need the community to understand that taking the vaccine is for their benefit. So unless, of course, the stance of the National Department of Health changes, that is already showing us that mandatory vaccine policies will not be a simple thing to just put in place in our workplaces. If you look at the Occupational Health and Safety Act, the employer actually has a responsibility within reasonable practicable means to provide for the health and safety of persons at work. And so that will include offering and probably capacitating or ensuring or optimizing coverage of COVID-19 vaccination within the workplaces. So again, just overall, although it may be the easy approach to force employees to be vaccinated, this may be possible in certain settings, but generally this will not be an easy to implement thing. So the net balance of the existing legislature does not allow for blanket mandatory vaccination policies. And in the absence of that, then where does this place us as occupational health practitioners? It really tells us that right now, in this preparation phase, we have to really be developing capacity within our employees to be able to make informed decisions about vaccinating, whether to vaccinate or not. And so this effort will really require multiple stakeholders to come together in order to facilitate and to capacitate these employees. The occupational health practitioner, of course, will be central to all these efforts, as they are really the link between management operations and the employer for a program such as this. But also, more importantly, employees and their labor representatives need to be engaged. And the way that they need to be engaged is really through communicating all the knowledge that Dr. Takuba has just taken us through, to get to a point where employees and the labor representatives actually understand very well the different vaccine candidates, how vaccines are developed, their safety profile, and to also start busting some of those myths that are really so predominant within our communities and workplaces. Management has to buy in, of course, because although some programs will be set in the context of the national rollout, there still needs to be support in terms of logistics and some financial support in terms of actually making sure that these workplace vaccination programs come into full play and are actually effective. And human resources as well will be important when thinking about people getting reactogenicity, scheduling days for coming to vaccinate, as people may have satellite work operations. You may want people to come to a central place, and people afterwards may need to take a day or two off when they are not feeling well. So all these stakeholders will really need to come together to make sure that employees are capacitated with the right information and also the correct and conducive environment in order for them to make decisions about vaccination. So in terms of, so essentially what we are saying is that this is all about overcoming vaccine hesitancy in our workplaces. It's about setting up a good program, but also overcoming vaccine hesitancy. And like I said, I think the steps to do that have really been built very beautifully in Dr. Takuba's presentation, because it's all about us as occupational health practitioners making sure that our employees understand the vaccine science in terms of how does the vaccine work and how is it developed. Already there are so many questions. How is it possible that we don't have a vaccine for HIV? And I work in the HIV vaccine space. So, but we managed to have a vaccine for COVID. in a few months. So understanding that no shortcuts were taken in the vaccine development phases, but people will also need to understand the vaccine efficacy or effectiveness, and this will also depend, this also varies and depends on the vaccine candidate. So employees will need to understand what the workplace is actually offering at that point, and what is the efficacy of that vaccine, the context in which the vaccine was trialed to for us to produce those numbers of efficacy, and also more importantly, the need to continue using non-pharmaceutical interventions even after people have been vaccinated in the workplace. Employees will also need to really understand the safety profiles of the various vaccines, a common reactogenicity profile of all vaccines, as Dr. Takuba has just taken us through. And we also need to really contextualize serious adverse events in the sense that these are rare and they are not as serious or as common as COVID deaths or other COVID complications that we have come to be so acquainted to in the past year or so. And also understanding the regulatory processes that take place in the development and licensing of vaccines. And more importantly, that no corners have really been cut in this process. So after really working on optimizing coverage for vaccines in the workplace, we will also need to really think about how do we manage exemptions, because the truth of the matter is that we will not be able to vaccinate each and every single person. And while the president has said a vaccine will be available for each and every single person, we're really targeting to vaccinate about 67%, around 70% of the population. So how are we then going to manage exemptions? And some of these exemptions could really be medical exemptions. So people who have severe allergic reactions to vaccine constituents. And these people, you don't want to call them into your small workplace clinic and vaccinate them. If they have a reaction, you need to make sure that probably you communicate with a specialist and they are vaccinated in a well-controlled area where if they have a reaction, everybody is geared to supporting them and all the medical intervention that may be required for this person. So these sort of people will be exempt from our workplace programs. But more importantly, we need to put in a contingency plan for them. It cannot be seen as us discriminating against people because of their prior medical history. There may also be religious exemptions, and we've seen quite a lot of this in social media. But I think the important thing here for occupational health practitioners is if the employee is willing, you may need to explore those grounds that they are basing their religious exemption or their right to be exempt for religious reasons on. So if we understand the vaccine science very well and what the constituents of the vaccines are and the development process, we may actually find that in this the base of this rationale to say I will be exempt for religious reasons is actually based on a myth. And we may be able to provide enough information for those people to then come on board and also be vaccinated. And the same really is for people who may choose to be exempted. As we said, currently, the legislative framework does not create space for mandatory vaccination for everybody. There may be certain instances where it may be where there may be the ability to push more for vaccination, but vaccinations, COVID vaccinations, have not been made mandatory in this country. So what do we do when someone chooses not to be vaccinated? And again, it goes back to understanding the vaccine science and the development and educating our employees accordingly. And when we've done all this, then it's really important to document the fact that this employee was offered a vaccine so that tomorrow, when more people are getting vaccinated and that employee cannot turn around and say, I was not offered an opportunity to be vaccinated and I was discriminated against. So documentation of the vaccine offer and the discussions that have taken place will be very important. And this documentation, importantly, should be in the employees' medical records. So it's not for public display. It's still part of their medical information. And also obviously making contingency plans to say that if some of our people do not take up vaccination with the first wave, a rollout or wave, can we accommodate them as we go into further rollouts, or will there be other opportunities for them to then be vaccinated? So when we come into the operational phase, I just want to talk about 3 things, and starting off with vaccine scheduling. So it's going to be very important when scheduling vaccination to balance between optimizing vaccine coverage and really, we will really be looking at vaccinating as many people as quickly as possible, so that we can then move into a phase where more workers are protected against COVID-19, and maybe operations can go back to normal, or this new normal, whatever it may be post-vaccination, and balancing that against the operational needs of continuing to work, as well as accommodating people who then need to be vaccinated. So we may be having to look into staggered approaches, and a staggered approach may look at mixing job categories and shifts in order to make sure that you vaccinate people, you give them an opportunity. There may be some people who will take a day or 2 off post-vaccination because of reactogenicity, or people who may not be as productive as they may usually be because maybe they have myalgia post-vaccination, and mixing job categories and shifts in a way that operations continue, but we also vaccinate as many employees as quickly as possible. And then finally, I've already spoken to the reactogenicity, considering the reactogenicity profile, especially in our national rollout now. I think it's the Pfizer vaccine that's coming in, and we know it's a 2-dose regimen. And actually what has been noted is that with the second dose, reactogenicity seems to be more enhanced. So then it's taking all those sort of factors into account. And then obviously, depending on the vaccine that will be available for your workplace program, you need to think about storage and distribution of the vaccine. So do we have reliable refrigerators and freezers? So for example, we know that the Pfizer product needs to be stored at ultra cold environments, that's minus 70 degrees. So that is, I don't think many people have that in their occupational health clinic. So already that means procurement processes have to take place. And in addition to that, all the cold chain needs to be monitored. We need monitoring devices. We need protocols on if we have temperature excursions in the fridge. How are we going to manage those vaccines? Are we going to quarantine them? How long are we going to contact the manufacturer, and what is the response that they're going to give us? How long will it take for us to get a new supply? So all those considerations now need to be built into a temperature excursion protocol or a vaccine storage and distribution protocol that we will need to have in our occupational health clinics. This will then also include, obviously, vaccine inventory management systems, and the fact that some people may choose not to bring all their employees to that particular occupational health clinic, but to actually go out into the satellite workplaces and vaccinate employees in those places. So how are we going to transport these vaccines at the appropriate temperatures to maintain the cold chain? And then the last thing that I want to talk about, which is really at the core of what Dr. Takuva and I do, which is safety monitoring. Then as occupational health care workers, we really have to think about how are we going to be reporting on adverse events following immunizations, because this forms a core component of pharmacovigilance. And is also going to be very important in terms of managing the messaging that's going back into the community where our employees come from about what are the adverse events that we are seeing, how are they being managed in a way to reassure the community and to have a level of transparency. So with the national rollout, Currently, the pharmacovigilance team is embedded into the expanded program of immunization, and all the reporting is aligned to that program. They have currently developed paper forms which are specifically for COVID, but if you look at the form, it's really 99% similar to the normal EPI case reporting and case investigation forms for AEFIs that we are accustomed to. And the National Department of Health is also currently working on moving this paper-based system to an electronic tool. And this will be very important because it will facilitate real-time reporting and management of adverse events as we come into the national rollout. And that is my final slide. Thank you very much, Ashraf. I'm going to hand back to you.
Thank you very much. That's the second of the tag team of the Sisonke Open Trial colleagues. That was Dr. Azwita Kalani, who had followed on what Dr. Simba Takuva has shared. They are both safety physicians as well as medical monitors at Sisonke Open Trial. And Dr. Takalani has just dealt with COVID-19 vaccine workplace program considerations. And it is so enlightening to hear from these two experts and with great experience in the field in giving guidance and direction to us in what we can do in the workplaces and what we need to prepare for. Thank you very much for that. We have slightly reshuffled our sequence in the program, and I have the pleasure now of calling upon Dr. Lumka Puwani, who is also in the trenches, boots on, dealing with the vaccination programs at Steve Biko Academic Hospital. And whilst Dr. Lumka Puwani, her presentations is going to be shared either by her directly or by my colleague Tabani in the IT department. By way of introduction, Dr. Lumka Puwani is an occupational medical practitioner at Steve Biko Academic Hospital. And in our first edition of this particular webinar on the 4th of March, she had to rush from her practical clinical engagements back to an electronic device to rejoin us. And we were very thankful for her taking time off during that particular webinar in joining us. Dr. Lumka Puwani is going to, as an occupational medical practitioner, give her perspective on the topic and occupational health-based clinical trial in vaccination. And we do know that the Steve Biko Academic Hospital is one of the official sites for the current jabs and the trial-based vaccines being made available to the frontline workers in particular in our public health sector. So I'm going to ask Dr. Puwani just to unmute her microphone and to check with my colleague Tabani if he could start. There we go. I can see the slides, if you could maximize that for us. Dr. Puwani, just to check your audio, could you unmute your microphone? And whilst we're doing that, a reminder, please, I've seen some attendees type their questions in the chat box. I'm trying to catch up with all of you to remind you to please go and paste your question in the Q&A box, the question answer box, where Dr. Takuva, Dr. Takalani, and Dr. Puwani has already been assisting, and Professor Elizabeth Main will be asking to also look at the questions there in order to get a sense of the trends of your questions and your needs. So yeah, that is Dr. Puwani joining us, and I'm going to just hand over to her. I've heard her microphone. There might be another device with the audio on in the background, Dr. Puwani, that has produced a little bit of feedback. If you could just check the other device as well. So please type your questions, copy it out of the chat box, and please type it in the Q&A box, because our colleagues have been answering your questions in the question and answer box. So I immediately hand over to Dr. Lumka Puwani. Thank you very much for taking time out of your very busy schedule. You are in an academic tertiary institution, hospital with academic connections as well. So we thank you for making time to join us. Please proceed.
Thank you so much, Ashraf. How's my audio now?
It is perfect. Please proceed. Thank you.
Thank you so much. Good morning, everybody. I am just going to take you through the process of our vaccine rollout here at Steve Biko. As we all know that we were one of the accredited sites for vaccination. And the first slide is basically on the reasons why we had to go about the rolling out and what led us from changing from the AstraZeneca vaccine to the J&J vaccination vaccine. We can move to the slide.
Dr. Bhavani, yes, Tabani just told me he needs to change your slide view to maximize it. He's just going to take a few seconds. He might need to close it and reopen it. Oh, there we go. Thank you very much, Tabani. Please proceed, Dr. Bhavani. Sorry for the interruption.
Okay, all right. Oh, as I was saying, the first slide is briefly a background on the reasons why we all had to vaccinate and the reasons why we had to change from the initial vaccine that was available for us, South Africa, the AstraZeneca, and change to the J&J vaccine. We all know that it was all because of the efficacy on the variant that is predominant in South Africa. So that one was not efficacious, and then we had to change then to the J&J. So when it came to planning, that is the rollout plan and the process flow, collaboration between the various elements and departments had to be involved. Let's go to the second slide. Yes, that is just the brief background. That, as we all know, that we then had to change from AstraZeneca to the J&J, which is basically a clinical trial because it was not approved. So it was at phase 3B clinical trial. And the initial planning was it had to involve 100,000 participants. But we all know now that we have exceeded the number of participants to way above that 100,000. And it included primarily healthcare workers from both public and the private sector, because the general population and those with comorbidities were supposed to be involved or in the second phase of our rollout plan, which was due to be now end March and April. So the clinical trial, as I've mentioned earlier, that it had to take place between the public sector, that is the Department of Health, and the Sisonke clinical trial. We can move to this next slide. Yes, so the clinical trial in our settings at Steve Biko was led and conducted by the research groups, such as the Medical Research Council, the SciNexus, and the Sichaba, which are all research groups. And this was under the Sisonke study. And then the next slide. So the process flow, we had to have initially a collaboration between the Department of Health, the research groups, and occupational health of Steve Biko in order to come up with all the plans and the process flow of various duties and tasks that had to be taken. And as we all know that our primary duty as occupational health is to promote health and safety amongst our healthcare workers. So the first step of collaboration or of the rollout plan, one, we had to identify the site as to where we're going to vaccinate, to the times when we are going to operate, and also the booking system what system are we going to use? So various departments within our facilities had to be involved. That is communication departments, the IT department, and infectious control department, and a buy-in as well from labor unions. We also had to come up with a plan as to how we're going to go about vaccinating our healthcare workers. So we had to group them according to their risk base. So the classification that we came up with here at Steve Biko was the one that is up on the slide, where we had to categorize or classify our healthcare workers according to their risk base. So we had category 1 up to category 4. Category 1 was basically those that are in contact, or the staff that is patient-facing. Patient-facing as in those that are dealing with the COVID-confirmed patients. So that is the staff that work in COVID wards, COVID tents, emergency units, theaters, those that incubate and ICU. Those ones, we had to prioritize them. So those were the ones to be in our first group of healthcare members or healthcare workers that were going to be vaccinated first, followed by the second group, which is category 2, those that are seeing patients that are confirmed and those that are suspected to be having COVID. And then category 3 is those ones like the ones that work in the pharmacy and the admin clerks. The list is those ones that we see there. And then with category 4, which is the last category, it's basically those ones that are not patient-facing. That is the classification or the category of our healthcare workers. The next slide. So after coming up with the plan and prioritizing our healthcare workers, communication was also very important now to spread the word to all of our healthcare workers, from the clinical departments to the support groups, of what we were planning to do and which employees were first to be, or all of them, which ones were then our plans. We had to just communicate to all of them the plan, the whole rollout plan, and communication played an important role to ensure that efficient internal communication and information sharing was conducted throughout the facility. And then the planning, which involved mainly identification of the site, booking system, security, because the vaccine had to be secured from the vaccination site up until it comes to, that is the preparation site, up until it comes to our site, that is at Steve Biko. And then the resource allocation as well, as to how many vaccinators are we going to need, how many nurses, how many runners, computers, printers, and all of those things. Those also were important in the collaboration, and also the vaccination itself. So those are the elements of collaboration that were involved. We can then move to the next slide. Before, or when we were ready with our plan, because we had to develop as occupational health the vaccination rollout plan, we then had to have webinars and team meetings before the onset of the rollout itself, that is the research itself. So the stakeholders that were involved was basically the Department of Health and the CEO of the hospital, occupational health practitioners, and the sisters and the whole unit of occupational health, the IT unit, communication department, pharmacy, admin and logistics, and the security. Those were all involved. And like I mentioned earlier, even the labor unions were also involved. the research units, which is Medical Research Council, Sisonke, and Synexus. We had meetings with them before the rollout plan. All right, we can move to the next slide. I think I've mentioned this one earlier on, that the Steve Biko occupational health played a very important role in educating and training and encouraging healthcare workers to enroll on the system, because as we all know, initially that people were very fearful of the vaccine. So we needed to educate them, train them, and encourage them to vaccinate. And I must say, since we've started and up until the end, majority of our staff members, they came out and they were all vaccinated. So they had to register on the EVDS system, which is the electronic vaccine system, for the rollout plan, where you had to give your information and where you were working, whether you are a frontliner, that is category 1 or category 4. And then the Sisonke as well, later on you had to register as well on the Sisonke research, where they would then give you a voucher number to say you are eligible to come at a given time and at a certain date. And also, after identifying the site, we had to communicate and give directions as well as to where the site is going to be, how you reach there, and the times, the operating times. And SMSs were sent to all staff members that were registered, that is with the EVTS system, and all of those who got the voucher numbers. They were sent SMS, they were sent SMSs as a confirmation that they've been booked and they were ready for vaccination. And then when we started rolling out, we had sessions on daily basis, 2 times, in the morning and in the afternoon. And later on, when there was a required need, we had to have briefing sessions or feedback sessions on the progress of the rollout plan, where we then reported about all the admin issues, the vaccine itself, the queues and queue management, the numbers of the runners that were available on the floor, and also issues if there were with our pre-booking systems. So those were the issues that we needed to give feedback on them on daily basis once we started with the rollout plan. We can move to the next one. That is the planning part, site identification and preparation, and the development of the consent forms, which in this case, Sisonke came up with a consent form where you had to fill in the consent form online and come on site with the voucher number and the consent form. And also the adverse event reporting tool. Initially, we prepared the adverse event tool for the first 2 to 3 days. And then later on, Sisonke came up with their adverse tool where we had to report each and every adverse event. As we all know that it is one of our duties as occupational health, doing medical surveillance and also the reporting of all the adverse events. So they were all reported on the tool. We also had the appointment system that is allocating all our healthcare workers on different slots in order to avoid long queues and long standing hours and long crowds at our vaccination site. So people came with the pre-booking slots for different time from the morning up until 5:30 when we close our site for vaccination. And then the next slide. With planning as well, security was of importance. That is making sure that the transportation from the Synexus, which was our storage site, we had to make sure that the vaccine or the IP was properly secured. So arrangements were made for the transportation and ensuring that from the site up until us, that is the vaccination site, it is properly secured. And also the availability of securities at our gates and the presence of the queue marshals who were managing the queues from our entrances up until our vaccination site. And information and technology, that is IT, also played a very important role in ensuring that the systems are up and running. The computers, the printers, and all the like, all of them were available. And the training of our vaccinators, we had to educate and train them before they could start vaccinating our healthcare workers. And then infection and control measures. We had to take a proper infection IPC measures at our vaccination site, ensuring that the area is well ventilated to prevent the spread of COVID-19, and making sure that hand hygiene is adhered to. So the basins and soaps were all available. And also distancing, social distancing, we had to make sure that, like I mentioned earlier, that there was a pre-booking system to avoid queues, and also to make sure that social distancing is allowed or is adhered to. We had to screen our healthcare workers on arrival at the vaccination site. So the COVID-19 screening was applied, and then they had to give us on registration the number, because we issued them with a number. Once you've been enrolled and registered on the EVDS and under the Sisonke, you came in with a voucher number and the number that we then gave you at registration, where you then had to wait after registration. You then, we double-checked all the necessary credentials, and you were given a number, and a consent form was also signed again at the registration site. And then you were then marshaled by the queue marshals to the vaccination area. The vaccination site, we had areas for vaccination and areas for observation, 15 minutes observation after vaccination. And thereafter, if nothing is observed or you do not present with anything, you would then exit the vaccination site. We can move to the next slide. Human resources also, like I mentioned, on the elements of collaboration, played an important role in making sure that we have enough vaccinators for the day, as we had targets for each day to say, today we'll be vaccinating, for instance, 500 healthcare workers. So we needed to have adequate number of vaccinators to be able to ensure that we reach our target. And the runners that were involved as well, making sure that they take the vaccine from the pharmacy to the vaccinators, and also from the vaccinators to the runners as well. And if any help or need was needed for the runners, they were all available, even with the registration, making sure that all the process flow runs smoothly. And then we had data capturers from the IT who were using computers, laptops, tablets, and routers, and the telephones on site. And then with the pharmacy, availability of fridges and cooler boxes, and the photocopying machines, IT made sure that they are available. We can move to the next one. All right. or like I mentioned, data was the mainstay of the research. So they had to collect all the information on real time. And then on site, data capturers from SPA and also the research group later on provided us with data capturers to see to it everything runs smoothly. Initially, on day 1 and day 2, we collected data on paper and on the computers, but later on, we completely changed to paperless collection. So no paper was used, it was mainly EVTS and the Sisonke online. So the data that was collected included the identification of the healthcare worker and recording of the vaccine, that is the IP dose batch number. And this was done by the vaccinators. And then on that record, the vaccinator will then write and initial his name and surname and the number of workers vaccinated. They will also write on that note. And then at the end of the day, we could then reconcile as to say in the morning, the target will be how many we are targeting for the day, how many doses are prepared for that day. And then at the end of the day, we'll then reconcile as to how many have been vaccinated on that day. So we tried by all means to avoid any form of wastages. So collaboration between the vaccinators, the runners, and the pharmacist was of crucial importance once the rollout plan was up and running, because each day we had to communicate with each other to say, how many have been booked for the day, how many are coming at such and such an hour, and how many they can prepare. So it was also very important, that one. You can move to the next one. And the vaccination, it was basically managed by us, occupational health, the Q marshals, the vaccinators, training, and making sure that at the end of this whole project, all our healthcare workers have been vaccinated. And as you can move to the next one. Yeah, okay. Before the issues of concern, maybe if I can just give a brief collection or report, since we all know that we started on the 17th of February vaccinating, and then we finished on the 17th of March. We finished on the 17th of March. There are numbers that we were targeting. We were targeting to vaccinate all of our employees, which is 4,000 employees, including our interns that are around 2,000, which then gives us a total of 6,000. But we also had medical students and also allied students who also had to be vaccinated, and the EMS members that also came to our site to be vaccinated. So initially, we made sure that all of our healthcare workers, all the categories from category 1 to category 4 are all vaccinated. And then later on, the private sector was also vaccinated as well at our site, and the rest of the Tshwane public healthcare workers were also vaccinated. So at the end of the vaccination, 3,500 healthcare workers of Steve Biko were vaccinated. That is out of the 4,000, with 1,250 of the interns and 700 of our students were all vaccinated, and which I think is a very good turnout number, because of those that were not vaccinated, one, it's because they had allergic reaction to vaccines previously, and others were on leave, and a few of them were pregnant, so they couldn't take the vaccine. very few of them still had at the end of the project some fear to the vaccine, so they did not come out. But the overall turnout was very significant, which was very good. And then the issues of concern during the rollout plan and of the issues that we were discussing or feedbacking on daily basis was one, the conflict management during the operation, which was not prepared for adequately. And also we had challenges with our booking systems, especially in the beginning, that is the first week and the second week, where the EVDS system was not giving the voucher number or the number or the SMS to say you have been registered. So we had challenges with that. So some of the healthcare workers had to come on site without the full registration. So our admin and IT had to assist those healthcare workers who had challenges on that one. And then the demand as well, later on when the whole population of Tshwane heard that we were rolling out, there was then a large demand, a large number of healthcare workers who came on site for vaccination. And at times we had shortage for the day of the IP. And then the larger numbers, they've put a strain on our infectious control measures. And there was one weekend where we just had to call SAPS to come and intervene because we couldn't manage the queues. And long working hours were also an issue to our vaccinators and our runners, which included we had to work weekends as well. And we then realized as well that some of our vaccinators were not properly trained, and of which we then had to train them or retrain them again. So those are the issues of concern. We can move to the next slide. Also, the next slide is the thank you. But summary, since we finished last week with the vaccination, because we have handed over now, our site has been closed from last week. It's MedForum that is vaccinating now, both private and some of our members, that is from Steve Biko and also from the general public sector around Tshwane that have not been vaccinated. So we're still sending a few of those that have been vaccinated. So if I can just say a brief summary on our experience as the site for rollout. Initially, we experienced challenges, as I've mentioned, but at the end of it all, learning on day to day on what to improve on, and also given the numbers that turned out and the numbers that we vaccinated, I must say that our plan or our rollout plan was quite a success, because all of our frontliners, especially category 1 and category 2, all of them were all vaccinated. So that is just our experience at Steve Biko Occupational Health in the rollout plan that we have encouraged. Thank you.
Thank you very much. That was Dr. Lumka Puwani on occupational health-based clinical trial in vaccination and occupational medical practitioners' perspective. I'm going to ask our colleague that is going to Dr. Tendo Mpapele, who's going to stand in for Dr. Sipho Senabe, to just give us a brief overview of Dr. Senabe's contribution. We are running slightly behind time, and I'm going to hand over to Dr. Mpapele immediately. My sense is that he's also an occupational medical practitioner with sort of in the trenches experience at Charlotte. I hand over to you immediately, sir. Please proceed.
Thank you so much. Am I audible? The slides are visible.
Yes, slides are visible and you're audible. Please proceed. Thanks.
Thank you so much. I'm going to be very brief. I'm just going to cover mostly in terms of the bigger overall rollout plan that the province has. My slides, are they moving on your side? I'm not seeing them move here.
Not yet. Let's
try that. Yeah, I'll just move it like that. Okay, just looking at the stats, we've got about 11,833 healthcare workers in the province that have been positive. We've also got an additional 3,449 healthcare workers in the private sector that are positive. As you can see in terms of distribution of the different categories, healthcare workers are definitely at the most risks, as the numbers clearly indicate. If we looked at those hospitalized, because healthcare workers are the most people infected, we can see here that Gauteng and KZN were amongst the most affected in terms of hospitalization, looking at just about under 2,000. Of these, 98 unfortunately passed away, 50 remain in hospital, about 20 possibly with very severe disease needed rehabilitation centers. After that, the distribution amongst healthcare workers is mostly the nursing staff, mainly because they make up the bulk of the healthcare system in every single hospital, and they have the most engagement with patients, nursing them on a regular basis, as opposed to other departments that just come in and out as and when required. Also worrying our cleaners who are responsible for sanitizing the services and so forth, but also amongst the large majority of those that found themselves hospitalized, and then to a less extent, medical practitioners, as well as other allied health. Then this is just the distribution of how the curve worked in terms of the infection of healthcare workers. This pattern follows almost exactly the one for the rest of the province. We see that peak that happened after the regulations were put down and were lifted. We saw quite a number of people being infected, peaking at about July. Then we went down by September, had more or less no infections November and December. And again, as the second wave started peaking throughout December into January, we saw quite a lot of numbers going up during what was probably even worse than the first one because of a more severe strain. Then this again, not going to go into it too much. It just looks at the distribution of those that passed away, those that got discharged alive, those that remained in hospital, and so forth. We can see ourselves in KZN were amongst the most affected in terms of healthcare workers. So the crux of the matter looks at the vaccine from a more broader perspective as to where we are in terms of this rollout phase. So the vaccine gets developed in the labs and so forth by the scientists that know what they're doing. We look at now animal trials, starting usually with rats, they've got a quicker metabolism, you can see results quicker, moving to primates, which then resemble humans more genetically. Then we start just a small group of people just to see now the safety. Does this person, after giving this vaccination, do they have any severe adverse effects that are causing us to have to stop whatever it is that we're doing and revisit the first process? Should that be safe, we then move on to a bigger people. This is just getting more different people to get an idea of that in different demographics. How would this affect people? Then we look at phase 2. Now it looks at a much bigger, about a couple of thousands of people now. We've been looking at the effectiveness of the vaccine. Like, okay, it is safe, but is it effective in doing what it needs to do, which is now to prevent severe disease or prevent people from contracting this virus. Once that is done, we move on to phase 3, which is now tens of thousands of people, now looking at different demographics, short, tall, looking at different races and so forth. Again, looking at side effects and also effectiveness of achieving the intended goal, which is to minimize or stop the spread of that particular virus. So that being said, phase 3 is further divided into 2 stages, A and B. And A is where we engage by is regulated, generating solid pattern during this time, going to get the virus or not. When I get the phase, which is B, which is an extended trial, while we're waiting to launch. So once they're expecting or waiting for approval, we extend the trial, we can continue vaccinating with this particular J&J vaccine going forward. Now, in terms of our rollout in the province, we're looking at 3 phases. Phase 1, which is currently underway, which is the healthcare workers. We've got a population of healthcare workers. This includes both public and private that need to be vaccinated as part of the first phase. Once this phase is completed, it is expected, or at least anticipated, that by the end of May, ideally, we should have been done with this particular phase, as there's a shipment of Pfizer vaccines to supplement the current already going J&J vaccines that's expected over the next few weeks. We'll then move on to phase 2. Phase 2 has different people or categories involved: essential workers, persons that congregate settlements, persons over 60, persons over the age of 18 with comorbidities. As we can see, the different numbers which these populations represent. As part of the Phase 2, the people that are intended to start with currently, to my understanding, is going to be the people above 60 years old. This is because they're at most risk for severe disease and death. In Gauteng, we've got 1.3 million of people that fall under this category. The plan is to vaccinate them as of May, the next about the ones that get the most sick, and they're the ones that burden the healthcare system. So if we can ensure that this particular population is not getting severe disease or not getting infected at all, we can really lessen the number of hospital admissions, lessen the number of people that die due to this. Some of the stats that I saw said, if this is successfully done, that before June we've vaccinated this particular category, we can save about 40 lives a day during those winter months. So once those are done, we will extend the program to the rest of the people that fall under this category: essential workers, police, and 18. The last phase will be the rest of the population, which consists of about 22.5 million of South Africans. This is everyone over the age of 18 that will achieve our target of 67% of the population vaccinated. Then this particular program of vaccination now had a lot of people that a lot of vaccination clinical trials. We're still part of a phase 3B trial. We'll be looking at the effects and efficiency of this particular vaccine in a specialized category of what we call healthcare workers. It also had to do with looking at the vaccination regulatory requirement of the ILO, who also responsible for classification of hazardous biological agents. So currently we had it classified as hazardous biological agent 4. 4 meaning that this is a severe disease, can cause severe disease to humans. It is a serious occupational hazard and is threatening to life, and without any effective prophylaxis or treatment. That's how COVID was. We know last year there was no effective treatments against it or vaccines. Now that we've started a vaccine program, should it be successful, it's going to be reclassified into 3. It still has the same description in terms of intensity. It causes severe human disease and a threat to life, but at least there's a effective prophylaxis or treatment against it. So once this program is successfully run, we'll be classifying COVID into category 3. And then obviously it also integrated our national vaccination rollout system, which is now started in the form of this particular trial. Then duties of the state employers and employees regarding this vaccination trial. Number 1 is also looking at our Bill of Rights and our Constitution, which protects the right of everyone here, looking at the security and the control of their own body, and also looking at now people should not be subjected to any medical scientific experiments without informed consent. As part of this trial, particularly, there's a very extensive consent that is signed, a lot of leaflets that give you the exact information that you need to know, efficacious out for it. Once the bigger rollout happens, the consent will still be done, but it will not be as extensive as the requirements of this particular clinic.
It sounds like your audio is a bit glitching there, Dr. Papele. Could you move closer to your microphone?
Trial. Then general duties of the and also safer work environment is. Then COVID-19 is an occupationally compensatable disease. So this looks at the infection itself. Those that have been infected in the workplace, specifically healthcare workers, have to be submitted to the compensation committee and have their case looked at as to where the origins of the disease is, and should they have severe disease with following comorbidities after obtaining it in the workplace, they should be appropriately compensated as is by law. Then in conclusion, the vaccination of employees as part of this phase 3B trial is at the same time an occupational health intervention to monitor exposure to hazardous biological agent in the workplace. And again, trying to reclassify this COVID-19 from 4 to B, as we have now effective prophylaxis against it. Ethical principles of informed consent to participate in the vaccination trial are very important. It is a legal obligation. It is part of our rights. Everyone's got the right to determine what happens with their body, regulations regarding this. Then management of adverse events and serious adverse events have to be managed by the compensation for occupational injuries and diseases, as well as part of the clinical trial and quality management, meaning that as occupational practitioners, we should make sure we notify all people that have any adverse events requiring any days off. And as part of the clinical trial, we must make sure that this is documented so that when they do publish these results, they are aware of the side effects and the more common things that occur during after inoculation. And the governance of occupational health programs and clinical trials presents opportunity to balance ethics, laws, occupational health, policy, program management, and impose a culture of collaboration and transdisciplinary interaction. That's about it from my side. It's a summary of what the broader picture of the vaccination rollout looks like. Thank you.
Thank you very much for that. That's Dr. Tendo Mpopele, who kindly stepped into the breach there for Dr. Sipho Sanabe. I'm now going to immediately hand over to Professor Elizabeth Main, and I'm hoping that, as with the last webinar we had on this topic, she had a good scan of the questions in our question and answer box, as well as dealing with some of the very, very common questions that she's come across in her experience. I hand over to you immediately, Prof. If you could maximize your slides, that's perfect. I hand over to you. And as by way of introduction, apologies if I could just quickly do that. There is a very long CV, which I can't go through in detail, but as a brief, that's the wrong document, apologies. So as a brief introduction, Professor Elizabeth Main is head of the academic department immunology, and so that is as part of the NHLS as well, and associate professor at the School of Pathology at the University of Witwatersrand. And it's in the capacity as the NHLS head of immunology based at Charlotte Maxeke Academic Hospital that she's joining us. I hand over to you, Prof.
Thank you very much, Ashraf. Can I do a sound check? Can everybody hear me? Can you hear me, Ashraf?
Your audio is perfect. Thank you very much. Please proceed.
Fantastic. So I think what I'm going to be giving is much more of an introductory lecture, so not as much of an operational lecture. And then at the end, I'm going to do some of the frequently asked questions. And I updated these actually this morning to see if I could answer some of the questions that I've been getting recently. All right, so just to remind everybody what a vaccine is any preparation which acts to induce immunity to a disease or a substance. And it's there to stimulate some kind of response. And it's been called the greatest public health intervention of all time. And this is a picture of the last patient to die of smallpox. And just to remind you that smallpox has been eliminated or actually eradicated. So what do we want to do when we vaccinate people? So there are 3 levels. The first is control. So that's reducing the incidence of disease within a geographical region or morbidity. And we'll talk about that with coronavirus just now. The second is reducing the incidence of disease to 0 in a specific geographical area. And then eradication, which is what we've done with smallpox, is eradicating the worldwide incidence to 0 so that no deliberate intervention is required. So we've stopped vaccinating against smallpox. And then how do we assess if a vaccine has worked? And this is going to be important because we're going to come to this just now. There are 3 things that people often talk about when they're talking about vaccines. The first is immunogenicity. And immunogenicity just means, does the vaccine cause the individual to have a response? So does it cause an immune response? That's not the same as efficacy. Now, efficacy is what we're very eager to monitor in terms of the J&J vaccine, because what we're saying there is, under ideal trial-like conditions, does the vaccine work? So does it prevent disease? And probably what you're interested in, and what probably the greater population is interested in, is effectiveness. And that is, if you vaccinate people in the general population, does it help to prevent disease? So does it work in the real world? There are a number of different vaccines. You can either have a subunit vaccine, for example, acellular pertussis, that's just a part of the pathogen. You have live attenuated vaccines, so the vaccine pathogen is alive, but it's been weakened, so it no longer causes disease, and that's like, for example, the BCG. We have killed organism vaccines, and then we have this new category, which is what we're talking about just about here, is DNA and RNA vaccines. Now, the issue with DNA vaccines is that viruses live inside the cells. And because they live inside the cells, you need to ensure that the immune response is targeted to destroy those infected cells. And that's why we try to introduce DNA from the virus into the cell so that the cell will kind of respond as if it was infected, because we think that's the right way to stimulate the appropriate immune response. I'm going to skip this slide just to point out that this coronavirus and the important antigen, the important part of the coronavirus we're going to be talking about is the spike protein, because this spike protein binds to the ACE2 receptor, and that ACE2 receptor is how the virus enters into the cell. So once the virus enters into the cell, it injects its nucleic acid into the cytoplasm, and that gets translated into protein, and that makes new viruses, which can then go on and infect new cells. So when we're talking about viral vector vaccines, what happens is we take a virus other than the coronavirus, like an adenovirus, which causes a cold, and we allow that virus to transport some of that nucleic acid into the cell. Okay, so mRNA vaccines for COVID have been rigorously tested for safety before anybody authorized them for use. And this mRNA technology that we're using is new, but it's not unknown. It's not something that we've never used before. And we've used it for things like cancer research, as well as for research in things like TB vaccines. The mRNA vaccine does not contain the live virus. It contains a part of the virus that causes infection. And mRNA from the vaccine does not enter the nucleus of the cell. So here's a little picture again. So here we've got an adenovirus. You can see here's the DNA. The DNA gets injected into the cell. It gets loaded onto a sort of carrier molecule called MHC class 1. And that will then be introduced to the immune system, so the immune system can respond to it. And this introduction is known as immune presentation, so immunological presentation. And again, importantly, both this DNA and RNA vaccines try to mimic natural infection so that we can induce the right kind of immunological response. So as everybody is aware, we have a Johnson & Johnson vaccine, and that's the one that's been rolled out. And this packages some of the spike protein, the mRNA, which encodes that spike protein, the instructions for making that spike protein inside an adenovirus. It's very stable, and it's a single dose regimen. How effective is it? So 100% effective have been the responses from, for example, this is from JAMA, in terms of preventing hospitalization and death. Some of the other vaccines which are on the market include the Moderna and the Pfizer vaccine. And the Moderna and the Pfizer vaccine are also being looked at in the South African context. The issue with Pfizer is that it requires, as my colleagues earlier said, Azwi and Simba, that it requires refrigeration at very low temperatures. So these may be some things that we look about later. You're all aware of the AstraZeneca vaccine. We've recently sold that into the AU because it is not as effective as our against our South African strain. Okay, so as we mentioned, there's been a significant amount of tracking of these vaccines. And so a lot of people are in the race to make them. Okay, so let's just talk about some of your frequently asked questions. So that's a little bit of the science behind the vaccine, but I'm sure that the questions you want answered are possibly more of the operational ones. This is the first question that I got. I actually got a call from a friend of mine who says, please can you let us know what it means to have a DNA or RNA vaccine. Does it mean that my genetics will be changed? Now, that's completely not true. So although there are some viruses that incorporate themselves into the nuclear DNA, these viruses like HIV are retroviruses. Coronavirus itself is not a retrovirus. So although it injects its RNA into the cell, it doesn't end up in the nucleus. So neither the vaccine nor the coronavirus itself will inject and incorporate itself into the cell. What about the side effects of the vaccines? So you have to be aware that the vaccine is there to activate the immune system. That's the whole point of it. And activating the immune system does give you some temporary side effects. And majority of these would be the kind of things you would have when you have flu. So a low-grade fever, some pain, often muscle aches, sometimes a low-grade, a little bit of a rash. And generally these were reported as mild. As Simba and as we have already said, severe allergic or anaphylactic responses are very uncommon. So less than 1 in a million or 5 in a million participants. We have a 1-dose regimen for J&J, although we are investigating a 2-dose regimen as well. And what we do know is that these side effects are more likely to occur if you have the 2nd dose of a vaccine. Okay, so I have heard that individuals get clots following the vaccine. So one of the issues that has been raised, especially regarding the AstraZeneca vaccine, is that people who get the vaccine tend to develop abnormal clotting profiles. Now, as I mentioned, the whole point of the vaccine is to stimulate inflammation, and inflammation we know is linked to an increased risk of having clots in your legs, for example. But although this is a theoretical possibility, the risk of getting an abnormal clot with severe coronavirus infection is very high. And so in terms of risk-benefit ratio, it's probably easier and better to get the vaccine. If an individual is pregnant, should they be vaccinated? Now, initially there was a theoretical discussion that maybe pregnant women should be left off the vaccine schedule. And certainly for our J&J vaccine, which is still under clinical trial, pregnancy is considered a contraindication. However, the WHO has recently reviewed its decision not to vaccinate pregnant women, and now the suggestion is that women should be vaccinated in the 2nd and 3rd trimester. There are a number of drugs that can interfere with a vaccine. People who are immunosuppressed theoretically won't make as good an immune response as other individuals. So, for example, corticosteroid use, pain medication, etc., could suppress the antiviral immune response to the vaccine. However, again, this is a theoretical more than an absolute risk, and certainly we would recommend people taking, for example, an anti-inflammatory agent if they have pain or muscle aching after they've received the vaccine. What is important to tell your vaccinator? Obviously, pregnancy and breastfeeding is important. Anything that could infect the immune system, anything that has resulted in a severe anaphylactic response, for example, to previous vaccines, people who are susceptible to bleeding, any active COVID infection, or if you've had another vaccine for SARS-CoV-2. And it's important to say that none of these are absolute contraindications. All of these are relative concerns, but in most cases, these people would still be vaccinated for SARS-CoV-2. How long does this protection last? And a lot of people say, so is this going to be a once-off thing, or are we going to have a vaccine every day? We're not currently sure how long the vaccine will give protection for, because we only have about 6 months of data, and it may be necessary to have this vaccine more than once. Some people have suggested that you can mix and match vaccines. Is this true? So although it's not currently recommended that we mix and match vaccines, so give one dose of one vaccine and then a second dose of another vaccine, there is preliminary data from the UK where they have been doing this kind of process to show that it might be efficacious, although, as I say, we're still not 100% sure. What about children and elderly people? So it's not recommended for patients under the age of 16, although they are starting adolescent trials in some countries. And so those adolescents we're still waiting for those results to come out. The important thing about children is that they're much less likely to have severe disease, and they are also much less likely to transmit because of changes in their receptor for the coronavirus. Has been some data that elderly patients may not respond as well to the vaccine, and certain European countries are making some recommendations that possibly those individuals are vaccinated later. But in South Africa, I think we've made a decision to vaccinate. This is a term which is being sort of bandied around quite a bit, herd immunity. Herd immunity means how many individuals in a population, so herd is like the population, need to be vaccinated in order to provide protection to other individuals in the population who either cannot or will not be vaccinated. And we say that's about 2/3 of the population. And obviously, once we've reached that level, the risk of transmission reduces significantly because you have kind of created a firewall in terms of transmission. How do we know if the vaccine has worked? And I've received a number of calls and comments from individuals about people who've had a vaccine, then they went to a pharmacy, for example, or they went to a lab, they had themselves tested and their antibody test was negative. And for that reason, they say they didn't get the real vaccine, they got somebody else's placebo or something else. Now, importantly, we know that not everybody who has got natural infection against COVID will make antibodies. And a lot of the antibody tests that we use at the moment measure antibodies against something called the nucleocapsid. Now, you'll remember when I started this talk, I said the majority of our vaccines contain the spike protein mRNA instructions, because that is the important one which blocks infection. So if you go to a pharmacy that does an N test, a test against the nucleocapsid, then you won't pick up vaccine-induced antibodies. So they're very important in patients who have had natural infection, but not necessarily in patients who are vaccinated. I have had COVID, and this I believe came from the previous president of the United States. Surely that means I'm immune and I won't need a vaccine. What we know about COVID, coronavirus infections generally, is that there is a risk of reinfection. So even if you had COVID before, you do have a risk of contracting COVID again. So neither vaccination nor having had coronavirus previously are 100% guaranteed that you will have immunity. It is important to also think that what we are trying to do with this vaccine is not to prevent you getting infected. It is to prevent you getting so sick, and it's also to prevent you from dying. So we need to manage the expectations of ourselves and our patients. So it's important to remember that vaccination significantly reduces the risk of severe disease and death. And I think this was already mentioned by Azwi, but I'm just going to say it again. No person can be forced to be vaccinated against if they do not want to. And I think the president made this very clear when he was announcing the vaccination, that we cannot be discriminated against in South Africa if we haven't been vaccinated, and we cannot force our employees currently as it stands without major suggestions of how we change things to receive the vaccine. So I'm going to stop there. I hope that was helpful, Ashraf, and I'm going to hand back to you.
Thank you very much. That was very, very helpful. And even after having had the jab, every time I listen to a webinar like this, I fully understand the importance of people understanding the COVID-19 vaccination programs in general. And this is another opportunity to inform people within the workplaces, all that have an occupational health and safety function, from those who are the HR to those who are representing the union, the health and safety reps, the sharpshooters, the compliance officers, and all the professionals who support our workplaces to ensure that we are COVID-19 not just prepared, but also ensure that prevention is happening there. Doctors, Prof, just one question as a follow-up. A colleague asked me a while back about pregnant workers or pregnant women. What's the guidance there at the moment, and what have we learned, and what do we still need to learn?
So Ashraf, as I mentioned in my talk, so pregnant women currently, because J&J is in a phase 3 clinical trial, I believe that pregnancy currently is not recommended for enrollment. However, having said that, from other centers across the world, recommendations are now currently from the WHO that we should be vaccinating colleagues in their second and third trimester of pregnancy. So vaccination, again, is always a risk for inflammation. So the risk is not zero, but we do need to protect pregnant women, and there is some evidence as well that protection could be conferred onto the newborn as well. So I think that as soon as we've managed to approve or authorize our vaccines, then I think pregnancy will definitely not be something that is contraindicated.
Well, thank you very much. And for me, that wraps it up. Professor Elizabeth Main, thank you very much. The NHLS Head of Division, HOD at Immunology at the Charlotte Matkeke Johannesburg Academic Hospital. Thank you very much, Prof, for taking the time and joining us. And we look forward to future updates, and ending off with that very key set of questions that you get to and sort of capture it all in your session as well. Thank you, Prof, for your contribution.
Thanks, Ashraf.
And then also to all of our other colleagues, as you've seen in the program, Dr. Simba Takuva, the lead safety physician, as well as medical monitor at Sisonke Open Trial, and his colleague, Dr. Azwi Takalani, the safety physician and a medical monitor also at the Sisonke Open Trial. Dr. Sipho Sanabe has been replaced kindly by Dr. Oops, and now I've just missed the name there. Mapele also from Charlotte, and then just before him was Dr. Lumka Powani, who's also one of the boots in the frontline and on the ground as occupational medical practitioner, having dealt with the occupational medical practitioner's perspective. And a reminder to everybody, thank you very much for your attendance. We will be circulating a link to all of you that will be asking you to give feedback on this particular webinar. It is based on the Red Cap platform, and one of the questions we ask you there is to please indicate what other topics you would like the NIH's COVID-19 webinar series to deal with, and for the consideration of our colleagues internally who decide on the topics going forward. And then just a reminder for those who do require the CPD points, a link to a ClassMark online test will be sent to you, and as per the requirements for CPD purposes, the test would have to be completed successfully for you to be able to qualify for the CPD certificate of attendance. A general certificate of attendance is circulated to everybody, but I have been reminding some attendees to change their sort of mobile and cell phone details that pop up as your name and surname. And if you could rename yourself while you are in the webinar, it'll be great, so that there's no issues with you receiving a certificate that doesn't contain your name and your surname. And before I thank the panelists from the NIH's occupational medicine section, as well as our IT colleague Tabani, who's done amazing stuff, which you don't necessarily see as attendees of this webinar in the background when the technical things don't go all that smoothly. But as a reminder, please visit our website. It is 0 rated by all of the mobile service providers and an internet service provider. You will therefore not be charged any data or cost when you visit the NIH website. That is www.nih.ac.za. There is a wealth of COVID-19 presentations of previous webinars, including this webinar's details will be there, the presentations, the recordings, and both audio and visual, and a host of other resources. I'm not sure if the COVID-19 vaccine and potentially, I think, long COVID posters and so on is there yet, but I do know that colleagues have been working on that, and those resources are also placed on the website. That is www.nih.ac.za. If you go into YouTube and you search for NIOH South Africa, that is NIOH South Africa, you will find the NIH's YouTube channel, where all of the video recordings of these webinars have been made available since March of 2020. So with that, I thank you all. I will play a few slides as I shut down my video and audio for you to get some of the details I've just mentioned. Again, thank you very much to all of our speakers and presenters, and particularly to our internal panelists, led by and guided by Dr. Nompumulelo Ndaba, who's currently heading the particular section, the occupational medicine section. Her colleague, Dr. Oded Volmink, apologies. And then there's Dr. Edward Sepirwa, Dr. I think I've missed out on some of the names, but I will certainly make a point of thanking them in person as well within the NIH building. And thank you very much. Goodbye. See you in our next webinar.